Bisphenol A is one of the highest volume chemicals produced worldwide. It's more than 4 billion kilograms each year, and more than 100 tons released into the atmosphere in the manufacture of bisphenol A. Bisphenol A is used in polycarbonate plastics that are used in the bottle, water bottles, and baby bottles, and medical device, and other products, and also in epoxy resins that are used as to coat metal products and food cans, bottle tops, water supply pipes, dental products, dental sealants, and so on. Bisphenol A is also one of the most studied chemicals. You see, this is the publications in the Medline since 1960 until yesterday. I made it, and you see, in the last 2 years, it was published about 1,500 papers on bisphenol A. Most of them on toxicological aspects of bisphenol A. Is certainly one of the most studied chemicals we have. This is because of its putative endocrine disruptors properties, as you will see. Bisphenol A has estrogenic properties, and this become more there was more attention on these problems after the Wingspread Conference in 1991, when this word endocrine disruptor, when this term endocrine disruptor was coined at that time. I don't know how familiar you are with the subject, but anyway, I will try. Endocrine disruption. And this is part of the statement of this conference, Wingspread Conference, when they warned that many chemicals introduced into the environment by human activity are capable of disrupting the endocrine systems of animals, including fish, wildlife, and humans. Endocrine disruption can be profound because of the crucial role hormones play in control development. You see here that the natural hormone estradiol is, and the D-ethylestylbestrol is a synthetic strong estrogenic substance, and bisphenol A that has some similarities with DES. The bisphenol A has a very weak estrogenic activity in vitro. This is the response of mouse uterine estrogen receptor. This is the binding. You see here the binding displacement by the natural ligand, that is estradiol, and here the bisphenol A. It's a very weak ligand. This man, this Frederick von Saal, was very much involved in the studies of bisphenol A. I will explain why. This von Saal and co-workers described in the mid-90s that what is very important for the attention on bisphenol A health problems, that the work with, he showed that exposure to D-ethylestylbestrol, a synthetic estrogen substance, during prenatal development of mice at very, very low levels cause in the adulthood enlargement of prostate gland. So mice exposed prenatally to diethylstilbestrol at very, very low dose had an enlargement of prostate gland in adulthood. And this effect was also has some unusual dose-effect relationship that's called inverted U dose-effect relationship. You see, the effect increase at low dose and decrease at high dose. This was a very important milestone in the endocrine disruption issue. And he also demonstrated that this happened also with the natural hormone is estradiol. He implanted mini pumps subcutaneously releasing estradiol during mouse pregnancy and also found the same kind of effect. Very low increase in the estradiol levels during pregnancy cause an enlargement of prostate gland in adulthood, and higher dose the effect decrease. This was a paper published in the PNAS in 1906. After this demonstration with estradiol and DES, Fonsal and co-workers showed that similar happens with bisphenol A. This is the effect of bisphenol A in the binding displacement. Here you see the bisphenol A. This is estradiol and bisphenol A that's much less potent in vitro, the estrogenic binding in vitro than the natural hormone. Here is the same with protein in the incubation medium. And here the same experiment he did with DES and natural estradiol with bisphenol A. And he found that very low dose of bisphenol A during pregnancy, the increase in prostate gland in adulthood. And Fonsal and co-workers conclude that their findings showed for the first time that fetal exposure to environmental relevant parts per billion dose of bisphenol A in the dose range currently being consumed by the people can alter the adult reproductive system in mice. This was a very, very important paper, had a very, very impact because it's very, very low. Environmental levels of bisphenol A could have an important effect in adulthood. But after this, about 9 years after, Anna Soto in the USA also showed that very low exposure to bisphenol A also altered the mammary gland development. Perinatal exposure to bisphenol A, this is the control, this is the bisphenol A exposed mice in the perinatal period. In adulthood, the branching of these ductus of the mammary gland was very much increased. The number of buds and branching of this duct. So it had an effect. Exposure in the perinatal period at very low dose had an effect persistent into adulthood. Here you have the relation with the dose. And Soto and co-workers conclude that perinatal exposure to environmental relevant dose of BPA results in persistent alterations in mammary gland morphogenesis, what is related to cancer in some way. So these two papers and some similar papers from the same group had a tremendous impact on the idea we had about safety of bisphenol A, because bisphenol A has very low acute toxicity and no other, I would say, remarkable toxicity. But these two papers had a strong impact on our idea about bisphenol A safety at very low exposure levels. Nonetheless, the two papers in rodents are controversial. There are many other papers. For instance, this from John Ashby. John Ashby is known by his contribution to genetic toxicology, but he moved to this field until he retired. He started working in AstraZeneca, and until his retirement he did more with endocrine disruption issue. And he didn't find any effect on prostate gland of bisphenol A. Also, several other studies. I will show some of them. This one is by Rochelle Tile. He works in a contract lab for the industry. And she did a very long study, a 3-generation reproductive study, bisphenol A in rats, and found no effect. Her conclusion is that BPA should not be considered as a selective reproductive toxicant based on results of this study. The same was repeated with mouse, a 2-generation reproductive study in CD1 mice, and no effect. There's a lot of controversy about these studies and many criticisms. One problem she had, the only one I found in her study, was that she used as a positive control estradiol by the oral route. And everybody knows that estradiol is not active by the oral route unless you give extremely high dose that cause some liver toxicity. So this is the only weakness I found in her study. Anyway, the problems are controversial. But there are another aspect of the problem is that it has also been argued that major metabolic difference between humans and primates and rodents preclude extrapolation of this data on BPA development toxicity to humans. On one side, you have controversial data on rodents. On the other side, it's also said that data on rodents cannot be extrapolated to humans because of some kinetic difference. Metabolic BPA, when given by the oral route, is directly conjugate to glycuronic acid in the liver. It's also conjugate to sulfate, but it's more to glucuronic acid and producing the glucuronide, monoglucuronide metabolite. Since BPA conjugation is very efficient, BPA undergoes a marked pre-systemic clearance of first-pass metabolism when exposure occurs by the oral route. When you are exposed by the oral route, most of bisphenol A is efficiently conjugate to glycuronic acid. So you find only the glucuronide metabolite, conjugate metabolite. This is what happened here. By the oral route goes to the portal vein and is conjugate and is excreted. Part of it, this conjugate, goes to the it can be eliminated by the kidney, through the urine, or through the bile. In this case, it goes to the intestines, and here it's broken again. The conjugate is broken and reabsorbed, and then you have enterohepatic cycling. Enterohepatic cycling, there are some difference between species concerning this excretion through bile or urine. You see, lower conjugates are excreted by urine, and higher molecular weight conjugate are excreted by bile. But the threshold is different in rats and men, in rats and primates. See, the threshold in rats is about 300, 400 Daltons. In humans is 500, 600 Daltons. That means, and the BPA monoglucuronide has about 404 Daltons. So in rats, it goes to the bile and gets enterohepatic cycling. In humans, it goes mainly to the urine. Here, what you have, conjugate metabolite. In rats, it goes to the bile and blood and feces, urine, and in human is mainly to the blood and urine. Because of this difference, the way it is excreted was commented by the European Food Agency (EFSA) in 2008, when they re-evaluated the toxicity data on Bisphenol-A, and they conclude that owing to this problem, owing to the enterohepatic cycling and decreased first pass metabolism of BPA in rodents, results in higher plasma levels of unconjugated BPA in rats compared to humans given the same dose. And this, they conclude that because of the enterohepatic cycling in rodents and not in humans, you could not extrapolate the data because you give longer to be eliminated in rats than in human. Exposure in rats would be higher than internal exposure to unconjugated BPA would be higher in rodents than humans. So findings in rodents would not be extrapolated to humans. That's the free is the unconjugated BPA. This was the argument to consider that no observed adverse effect or lateral effect in rats, and using the uncertainty factor of 100 could be considered as conservative for humans. So they reduce the safety uncertainty factor for extrapolation for animal data for human. This is a very controversial decision, and many problems about that. The EFSA statement, however, that decreased first pass metabolism of BPA in rodents results in higher plasma level of unconjugated BPA in rats compared to humans given the same dose remains to be demonstrated. One thing, they mix it, but one thing is to have the enterohepatic cycling. The other thing as to have a decreased first pass metabolism. See? You can get the conjugate, go to the bile, to the intestines, is broken, go to the liver again, portal vein, and it's conjugated again, and this does not necessarily mean that you have unconjugated levels lessening in the circulation. And this can be demonstrated that the pre-systemic clearance in rodent is very efficient as well by several papers. I will show some data by Potting and co-workers published in 2000. Here you have different routes of exposure, rats, female rats. You have the oral route, the BPA, unconjugated BPA. It disappears very fast from the blood. When you give by the intraperitoneal route, it lasts longer, and subcutaneous route also. Because here you have a strong, the first pass effect, and in the other two cases, all part of administered dose goes to the liver at each heart cycle. And here what you have, the data of Potting. Here the oral route, male, the lowest dose, you have no levels detectable, you have no maximum concentration, no area under curve of blood levels. That's different if you give by the intraperitoneal route or subcutaneous route. However, this was made with C14 BPA. If you measure the total radioactivity, you will find something, because this is not the unconjugated bisphenol, is the conjugated metabolite that you are measuring. This is what I said. According to Potting and co-workers, C14 BPA derived radioactivity in rat plasma, time to not quantifiable levels after oral administration, not the BPA terminal elimination half-life, takes 72 hours. Time for attaining not detected plasma level of BPA after oral administration to rats were 0.083 to 0.75 hours in males and 1 hour to non-determinant in females. That is, BPA terminal half-life in rat plasma is like to be very short too. Also supporting the EFSA argument are data obtained by Foucault and Decan in humans. He uses, because they are very controversial aspects of BPA measurement in blood and urine. The possibility of external contamination is very high. So they use, to avoid this problem, you have two problems in analytical determination of BPA, unconjugated BPA. One is external contamination, because BPA is everywhere. And the second is if you have the conjugate during the process of extraction, you break the conjugate and get again the unconjugated unconjugated BPA. And that's the problem you have to control to have a good analytical procedure. And he used the deuterium labeler BPA. In this case, you avoid external contamination, because deuterium BPA is not found as an environmental contaminant. He didn't find unconjugated BPA in urine and in blood samples. He found only the glucuronide BPA in the urine. And the half-life was about less than 6 hours. Take together the kinetic data from humans and rodents, rats, suggests that after oral administration of equivalent dose of BPA, elimination of BPA metabolite, the monoglucuronides, from rat plasma is slower than elimination for human plasma. This is consistent with the occurrence of enterohepatic recirculation of BPA in rats, but not in humans. That is no doubt about that problem. But plasma levels of unconjugated BPA are extremely low in rats and humans, suggest that a marked pre-systemic clearance occurs in both species. So, based on the existing kinetic data, it's not possible to conclude that the difference of biliary excretion between rodents and humans result in higher plasma levels of unconjugated BPA in rats compared to humans, give the same dose. It's more or less the same. And this was very recently nicely demonstrated by this paper by Fonsal and other co-workers. It has just been published. It's in the environmental health perspective. It's in press. They compare. These are the levels in monkey, rhesus monkey, Macaca mulata, and in the mouse. The same dose, 400 microgram per kilogram. Added to the food, so by the oral route, rats, mice, and monkeys were exposed through the oral route, and you have the same levels of serum unconjugated BPA levels, exactly the same. There's no difference. They found unconjugated BPA on both species, a primate and mice, and no difference. And also, the measurement was different. In the monkeys, they use the deuterium labeled BPA, so there is no possibility of external contamination in this case. This was not possible in the mouse because the volume of blood is very, very small, so they use a more sensitive method, that is the tritium labeled BPA. There are no direct, one thing is that you have the enterohepatic cycling. The other thing is the efficiency of the pre-systemic clearance. There is no direct comparison between humans and rodents concerning the activity of these conjugation enzymes and the efficiency of this process. The only study I found in the literature was published by John Ashby and this group in, I think it was in 2001. Very they found that the maximum V maximum for BPA glucuronidation is higher in humans than in rats. I will show you that here. Here you have. You see, this is the chromatogram. They incubate bisphenol-A with microsome from humans and from rats, and found here you have the glucuronide BPA and the BPA with a longer retention time, and here you have the measurement of the metabolite, the conjugate metabolite formed by in vitro incubation with rat immature females, microsomes. You have much high production of glucuronide than human and man and female. That's much less. This would suggest that efficiency of glucuronide conjugated reactions are rats are much more efficient than humans in the conjugate of BPA. And here a very nice experiment when they use the yeast histogenicity assay. Here is the response of the yeast assay to the estrogen, and here when you add the BPA without this cofactor for the glucuronidation, UDPGA, that is, the BPA was not, the conjugate metabolite was not formed. So this is unconjugated BPA only. And when you add the cofactor, you start the conjugate reaction. So here, in this situation, you have both the conjugated metabolite and the unconjugated BPA, and then it goes to the right because the conjugate metabolite has almost no estrogenic activity. So the curve shifts to the right, and the same in the rat. The shift is even larger. This data indicate that humans are likely to be more susceptible to BPA-induced estrogenic effect than rats. As far as we're aware, there is no other study making a direct comparison between rodents and humans regarding the liver capacity to convert BPA into its glucuronide metabolite. With this data, we have some questions about the toxic dynamics and toxic kinetic of BPA. That's question are not answered, but anyway, what we think, hypothesis to explain the whole thing. Could circulating BPA conjugated be cleaved back to BPA in the target tissue, for instance, by locally existing big glucuronidase? There are some studies that suggest that this possibility, this cleavage may occur at the target tissue. So you have in the circulation, systemic circulation, the conjugate metabolite, but at the target tissue, it's cleaved back to BPA that has some estrogenic activity. The other possibility, does BPA modulate expression and activity of enzymes and transporters involved in estradiol metabolism in the liver? This is also possible. So one possibility to explain the effects, in vivo effects of BPA. is not direct, an action direct in the receptor, but an action, an interference with the natural estrogen metabolism in the liver. And a third possibility, could BPA conjugate be hormonally active by mechanism other than a direct stimulation of estrogen receptor? A possibility that remains to be better investigated. Concerning the exposure, the internal exposure to BPA, contrasting with this paper by Foucault and co-workers, we have recently published Environmental Health Perspective, a review on the biomonitoring studies on BPA. There are more than 8. We found more than 8 biomonitoring studies on BPA, including thousands of individuals from several countries and as different methods. I'll show, if somebody is interested, can look at the paper. But the different population, adults, adolescents, children, and unconjugated BPAs was routinely detected in blood in the nanogram per milliliter range, and conjugated BPA was routinely detected in the vast majority of urine samples. Of course, in urine, more often we found only the conjugate metabolite, but in the blood, there are fewer studies in the blood, but you have consistently detected also the unconjugated metabolite in the blood. And it's our conclusion. Available data from biomonitoring studies clearly indicate that the general population is exposed to BPA and is at risk for internal exposure to unconjugated BPA. The two toxikinetic studies that suggest that human BPA exposure is negligible have significant deficiencies, are directly contradicted by hypothesis-driven studies and are therefore not reliable for risk assessment purposes. Oh, everybody is wrong and Foucault is right. What? The opposite. This is just one table. I will not go into details, but you have here, you see, different methods, GCMS, HPLC, and many different. We discuss all those possibilities in this review. And here, the most, I show you some more recent publication on this. This is a paper published in the American Journal of Medicine, Journal of American Medical Association. That was published in 2008 using data produced by a large study by the CDC. This epidemiology study found an association between urinary Bisphenol-A concentration with several diseases. Here you have the summary of the data. The several diseases here are arthritis. There's no difference between control, not exposed and exposed. Cancer, no difference. Cardiovascular disease is not significant. There is a tendency, but it's not significant. Angina is significant. Coronary heart disease is significant. So, people with a diagnosis of coronary heart disease are more exposed to bisphenol A. The same for diabetes. Say they found a significant association between levels of bisphenol A in urine and this diabetes and coronary heart disease. This is a strange association because you see, bisphenol A is not persistent in the body. It is eliminated, and what you have here, and the disease, coronary heart disease and diabetes, starts much earlier in life than this study. So this is strange, and there are some possibility this is a spurious association. There is no cause effect, but it was found, and they repeat the study later when a more recent study, you see, and they again found for coronary heart disease a significant association. And when they pulled the data from the previous study with this one, they found most of these conditions, coronary heart disease, diabetes, to have a significant association with bisphenol A levels in urine and this disease. It's difficult to explain because, as I told, this is current levels of bisphenol A, and the disease began much earlier in life than this moment you are evaluate the exposure, but this is there. And this, after that, the number of the papers have been published in bisphenol A, almost 6,000 papers in bisphenol A, and we still have this. What's the internal exposure to bisphenol A? What is the hazard? Because these studies have not been reproduced. And consequently, what is the risk? That's what I have. If you have any question.
Very interesting talk. I think that the issue of contradictory evidence, mainly in papers coming from the industry, seems pretty common because the atrazine is more or less the same. So I wonder from you a reflection of this. The second one is a question actually. It's related to the regulatory status of bisphenol A here in Brazil. If you are thinking that, like in Canada, for instance, moving towards the elimination of the use of BPA in plastics, or
the first part of your question is about the I didn't get okay, that's that. Yeah. Focal was supported by the industry, the plastic industry, and that's one of the problems we have. Where is the objectivity of science? One side you have Fonsal. Fonsal was a very Fonsal is a well-known endocrinologist. In the 70s and early 80s, he became famous by his studies on influence of uterine environment in the sexual behavior in adulthood, and he showed that, for instance, a male between two females in uterus, in this polytoicos animals, the brain development is influenced by this hormone environment, and the sexual dimorphic behaviors in adulthood are affected. So males are feminilized, and this was very, very nice study. And he became involved with the bisphenol A in the middle 60s, and since then is only concerned with this problem. And he's a very, very, very conflict with the industries, with Ashby, with the people from the industry, and we don't know who is right. Okay, because it has become a very emotional subject. If you see, when you think that in the last 2 years we have 1,500 papers published, it looks like it's on the rise. It's increasing the number of studies, and we do not. It sometimes is very complicated to reproduce this study. You see, there's some very complex studies. You expose very early in the prenatal, perinatal period, and then you have to control the phytoestrogens because very low levels. This is the problem, low dose effects. You see, is the reproducibility of this low dose effect findings is a complicated subject. And in this case, it's even more complicated. You have to control the phytoestrogens in the diet you feed the animals. You have many things to control. This experiment by Ana de Soto, nobody has tried to reproduce this experiment because, you see, it's a very time-consuming and very complicated to study the branching of mammary gland ductus, and it's complicated. And so you have this conflict of data, and you have more. I have some additional data I didn't show that looks like more interesting if you are interested here. This is also very interesting paper. Is the effect of it's an experiment in rat, I think. About the effect of bisphenol-A exposure on glucose tolerance. You see, you have here bisphenol-A, and you see here is a glycemia. When you give glucose to the animals, and you have a rise in the glycemia. The estrogen increase, so induce insulin tolerance, so levels of glycemia are high than in the control. And the same with bisphenol-A given by the oral route. This is, you give his control, his bisphenol-A, the blue one, and here is the same effect with the estradiol. It should be by parenteral route here, but here is oral route. And here is you give insulin, decrease with estradiol, attenuate the effect of insulin. It induce insulin tolerance, and the same the BPA by subcutaneous route. And here, when you give BPA by the oral route, this is the control, you have an even higher effect. This is plausible because when you give by the oral route, the problem here is in the liver, and it goes direct to the liver. And this illustrates that some of these effects are plausible, and there are many studies of different effects of bisphenol-A and very low dose level. So we don't know. There's a controversy. There is this problem. On one side you have the industry, negative data. I don't know how data they really have, because they only publish what they are, what support their view. They don't publish data that are against. And probably on the other side too. If you do not find what you expect, you don't publish. And that is the problem. And the other question you have is about the in Brazil, I don't know, I think Anvisa has not, you know something? No, I don't think. I think environment, Professor Jardine has some in the water, but human exposure we have nothing. I am really interested in having some data on human exposure in Brazil. I'm really particularly in one thing that we are very, very interested is the exposure of the fetus. So we are very, very interested in measuring in the umbilical cord blood to have an idea, because this is also controversial, and you have some indication. You see, most of the effects that are cause for concern were detected with perinatal exposure, and I have very, very few studies in the world about exposure, fetal exposure or perinatal exposure. And we have some problems because most of the studies were with baby in the intensive care units. It's very high, but in intensive care units they use many plastic materials, and that's the problem.